The intersection of systems biology and synthetic biology marks a transformative era in the life sciences, reshaping our understanding and engineering of living systems. Systems biology, with its holistic approach to deciphering the complex interactions within biological networks, provides an essential foundation for synthetic biology, which seeks to design and construct new biological parts, devices, and systems, or redesign existing ones for useful purposes. The synergy between these disciplines has accelerated the pace of innovation, enabling unprecedented manipulation of biological systems for applications ranging from medicine and biotechnology to environmental sustainability. Systems biology emerged as a response to the limitations of reductionist approaches that focused on individual genes or proteins in isolation. Instead, it emphasizes the study of biological entities as integrated networks, where the behavior of the whole cannot always be predicted by examining the parts independently. This paradigm shift was catalyzed by advances in high-throughput technologies, such as genomics, transcriptomics, proteomics, and metabolomics, which generate vast data sets capturing the dynamic states of cells under various conditions. Computational modeling and bioinformatics play central roles in systems biology, allowing researchers to interpret these data sets, map out intricate regulatory circuits, and predict system-level responses to perturbations. Synthetic biology, on the other hand, draws inspiration from engineering principles, aiming to make biology easier to program and control. Its core methodologies include the standardization of genetic parts, modular assembly of biological circuits, and rational design of entire pathways or organisms. However, the inherent complexity and unpredictability of living systems often pose significant challenges to synthetic biologists. Biological components rarely behave in a strictly modular or predictable fashion due to context-dependent effects, feedback mechanisms, and emergent properties arising from network interactions. It is precisely here that systems biology becomes indispensable to synthetic biology. By providing quantitative models of biological networks, systems biology offers the predictive power necessary to guide the design of synthetic constructs. Through iterative cycles of modeling, experimentation, and refinement, a hallmark of systems engineering, synthetic biologists can anticipate potential pitfalls, optimize circuit performance, and achieve desired phenotypes with greater reliability. This interplay not only enhances the efficiency of the design-build-test-learn cycle, but also expands the scope of what is technically feasible in synthetic biology. One of the most profound contributions of systems biology to synthetic biology is the elucidation of gene regulatory networks, GRNs. These networks govern the expression of genes in response to internal and external cues, orchestrating complex cellular behaviors, such as differentiation, adaptation, and homeostasis. By mapping out the topology and dynamics of GRNs, systems biologists provide blueprints that synthetic biologists can repurpose or rewire. For instance, synthetic toggle switches, oscillators, and logic gates, fundamental building blocks of synthetic gene circuits, are often inspired by motifs discovered in natural regulatory networks. Mathematical modeling of these motifs enables precise tuning of circuit parameters, ensuring robust function even in the face of biological noise and variability. Metabolic engineering, a subfield of synthetic biology focused on optimizing cellular production of valuable compounds, also benefits immensely from systems biology. Cellular metabolism encompasses thousands of interconnected reactions, regulated at multiple levels and influenced by environmental conditions. Traditional metabolic engineering approaches, which relied on overexpressing or knocking out individual genes, frequently encountered bottlenecks and unintended side effects due to the system's complexity. Systems biology addresses these challenges by constructing genome-scale metabolic models, GEMs, that integrate omics data and simulate flux distributions throughout the network. These models allow researchers to identify rate-limiting steps, predict the impact of genetic modifications, and design optimal intervention strategies. The result is more efficient production of biofuels, pharmaceuticals, and specialty chemicals via engineered microorganisms. Moreover, systems biology provides essential tools for characterizing and mitigating the context dependence of synthetic constructs. When a synthetic circuit is introduced into a host organism, it interacts with native cellular machinery in ways that are often unpredictable. Resource competition, crosstalk with endogenous pathways, and unanticipated feedback loops can compromise circuit function. Systems-level analyses such as transcriptomic profiling and proteome-wide interaction mapping reveal how the host responds to the synthetic construct and vice versa. This information guides the redesign of circuits to minimize interference, enhance orthogonality, and improve overall system stability. The iterative, data-driven nature of systems biology has also transformed the process of synthetic circuit optimization. Rather than relying solely on trial and error, synthetic biologists now employ model-guided design, leveraging computational simulations to explore vast parameter spaces and prioritize experimental validation. Machine learning algorithms, trained on large data sets generated by high-throughput screening, further accelerate the identification of optimal genetic configurations. This convergence of systems biology and artificial intelligence is ushering in a new era of smart synthetic biology, where predictive models continuously learn from experimental outcomes to refine designs in real time. In addition to facilitating the engineering of single-cell systems, systems biology is vital for the development of multicellular synthetic biology. Natural multicellular organisms rely on intricate communication networks, mediated by signaling molecules, hormones, and direct cell-cell contacts, to coordinate development, differentiation, and tissue homeostasis. Synthetic biologists seek to emulate and expand upon these capabilities, programming populations of cells to perform distributed tasks, form spatial patterns, or self-organize into complex structures. Achieving such feats requires a deep understanding of intercellular signaling networks, stochastic gene expression, and spatial dynamics, all of which are core concerns of systems biology. Agent-based modeling, reaction diffusion simulations, and network analysis tools developed in systems biology are thus instrumental in designing and controlling synthetic multicellular systems. Furthermore, systems biology enables the rational design of biosensors and diagnostic devices, key applications of synthetic biology in healthcare and environmental monitoring. Biosensors typically consist of synthetic genetic circuits that detect specific molecular signals and produce measurable outputs, such as fluorescence or color change. Designing sensitive and specific biosensors requires knowledge of signal transduction pathways, noise filtering mechanisms, and dynamic range optimization, all areas where systems biology provides critical insights. By modeling the kinetics and regulation of sensor components, systems biology helps synthetic biologists fine-tune device performance, reduce false positives or negatives, and ensure reliable operation in complex biological environments. The integration of systems biology into synthetic biology also fosters the emergence of novel therapeutic strategies. One prominent example is the development of engineered cell therapies, such as CAR T cells for cancer treatment. These therapies involve reprogramming immune cells with synthetic receptors that recognize and attack malignant cells. Systems biology contributes by elucidating the signaling networks governing immune cell activation, exhaustion, and memory formation, informing the design of synthetic receptors and regulatory modules that maximize efficacy while minimizing adverse effects. Beyond human health, the alliance of systems and synthetic biology holds promise for addressing pressing challenges in agriculture, energy, and environmental sustainability. Engineered microbes are being deployed to degrade pollutants, fix atmospheric nitrogen, or sequester carbon dioxide, contributing to ecological remediation and climate change mitigation. Systems biology provides the comprehensive understanding of microbial communities and metabolic networks necessary to optimize these interventions and ensure their safety and effectiveness. In agriculture, synthetic biology is enabling the creation of crops with enhanced yield, nutritional content, or resistance to pests and diseases. Systems biology informs these efforts by mapping plant signaling pathways, stress responses, and gene-environment interactions, guiding the rational engineering of desirable traits. The collaborative ethos fostered by systems biology is also shaping the culture of synthetic biology. Both fields emphasize interdisciplinary teamwork, bringing together biologists, engineers, computer scientists, mathematicians, and physicists to tackle complex problems. This convergence has led to the development of shared standards, open-source tools, and community-driven repositories of genetic parts and models. Initiatives such as the Synthetic Biology Open Language, SBOL, and the Biomodels Database exemplify the commitment to reproducibility, transparency, and knowledge sharing that underpins progress in both domains. Looking ahead, the future of synthetic biology will be increasingly shaped by advances in systems biology. The advent of single-cell omics, spatial transcriptomics, and real-time imaging technologies promises to deliver unprecedented resolution in mapping biological networks. Integration of multi-omics datasets will enable more comprehensive and predictive models, capturing the interplay between genetics, epigenetics, metabolism, and environmental influences. Advances in genome editing, such as CRISPR-based technologies, will facilitate the precise manipulation of complex networks, informed by systems-level insights. The rise of artificial intelligence and machine learning is poised to further revolutionize the interface between systems and synthetic biology. AI-driven models can uncover hidden patterns in large data sets, predict emergent behaviors, and suggest novel design strategies beyond human intuition. Coupled with automated laboratory platforms, robotic workstations, microfluidics, and high-throughput screening, these capabilities will enable closed-loop, self-optimizing systems for rapid prototyping and deployment of synthetic biological solutions. The societal implications of advances at the system-synthetic biology interface are profound. Engineered organisms have the potential to transform industries, improve public health, and address environmental crises, but they also raise concerns about biosafety, biosecurity, and unintended consequences. Systems biology offers frameworks for risk assessment, modeling the potential impacts of synthetic organisms on ecosystems and human health. Transparent communication, stakeholder engagement, and robust regulatory oversight will be crucial to ensuring the responsible development and deployment of synthetic biological technologies. The rapid evolution of both fields is also driving the development of new experimental and analytical platforms. High-throughput screening, single-cell analysis, and real-time monitoring technologies are generating ever larger and more detailed datasets. Systems biology provides the statistical and computational frameworks necessary to extract meaningful insights from these data, while synthetic biology supplies the tools to test and implement hypotheses in living systems. This virtuous cycle of data generation, analysis, and experimentation is accelerating the pace of discovery and enabling more ambitious engineering goals. Collaboration between systems and synthetic biologists is further facilitated by the development of standardized languages and protocols for describing biological parts, circuits, and models. Initiatives such as SBOL and the Registry of Standard Biological Parts promote interoperability, reproducibility, and open sharing of resources. These standards are critical for scaling up synthetic biology from small-scale academic projects to large-scale industrial and clinical applications, where reliability and consistency are paramount. As we look to the future, the boundaries between systems biology and synthetic biology will continue to blur. Advances in genome synthesis, for example, are making it possible to design and build entire chromosomes or even minimal genomes from scratch. Achieving functional, stable synthetic organisms at this scale demands an integrated understanding of the systems-level interactions that sustain life. Systems biology will be essential in modeling the emergent properties of synthetic genomes, predicting their behavior, and troubleshooting unforeseen issues that arise during construction and operation. The potential of the systems synthetic biology partnership extends to the realm of personalized medicine. By integrating patient-specific omics data with systems-level models of disease pathways, it is becoming possible to design tailored therapeutic interventions, such as gene therapies, engineered probiotics, or immune cell therapies that are optimized for individual patients. Synthetic biology provides the toolkit for constructing these bespoke interventions, while systems biology ensures that they are grounded in a comprehensive understanding of the patient's unique biology. In conclusion, the role of systems biology and synthetic biology is both foundational and transformative. By illuminating the complexity of living systems, providing quantitative tools for modeling and prediction, and fostering a culture of interdisciplinary collaboration, systems biology has enabled synthetic biology to move from proof-of-concept demonstrations to real-world solutions with profound societal impact. The ongoing integration of these fields promises to unlock new frontiers in biotechnology, medicine, and sustainability, empowering us to engineer life with unprecedented precision and purpose. As we stand on the cusp of a new era in the life sciences, the partnership between systems and synthetic biology will remain a driving force for discovery, innovation, and the responsible stewardship of biological technology.